All right, now let's continue with macromolecules 2. And we did discuss this in class, but when you look at these fruits, why did I say that some of you guys look like these fruits? Because the same DNA found in these fruits is going to be the same DNA found in us. We're talking now about nucleic acids. Our nucleic acids store and transmit hereditary or genetic information. Now your nucleic acids are your macromolecules that contain the 4 basic elements of life: hydrogen, oxygen, nitrogen, and carbon, and phosphorus. Okay? Your nucleic acids are basically polymers from individual monomers, and these monomers are what we're going to refer to as nucleotides. So one component, one monomer of a nucleic acid is what we refer to as a nucleotide. Now these are our 5 nucleic acids. Now, yeah, there are 5 because these 2 are interchangeable, where this one, uracil, is in RNA and thymine is in DNA. Okay, but they're both obviously not going to be found in the same place. They're both still nucleic acids. These are the structures of our 5 nucleic acids. Guanine goes with cytosine, and adenine goes with uracil in RNA, and adenine goes with thymine in DNA. So, here's our nucleic acid, our nucleotide. Our nucleotide has 3 basic parts to it. You have your 5-carbon sugar. You have a phosphate group on one end always, and you're always going to have a nitrogenous base on the other. Nucleotides, like for example ATP, play an important role in what? In capturing and transferring chemical energy. Your nucleotides are going to capture energy with your phosphate group, your nitrogenous base. We're talking about a 5-carbon sugar. So remember, you have sugar, ribose and deoxyribose, inside of your nucleic acids. And they help to transfer chemical energy, which then promotes also transferring genetic material as well. Individual nucleotides are going to be joined together covalently through covalent bonds, and they're going to form what's called a polynucleotide. We refer to a polynucleotide as a nucleic acid. They transfer genetic material, like I said, from one to the next, from one generation to the next. We have 2 basic types of nucleic acids. We have our DNA, deoxyribonucleic acid, and we have our RNA, ribonucleic acid. The major difference between DNA and RNA is very simple. 1 less oxygen. DNA has 1 less oxygen than RNA does. So as you can see, the component here in this example, you have 5 carbon, 10 hydrogen. Well, let me start here. In ribose, in RNA, you have 5 carbon, 10 hydrogen, 5 oxygen. DNA is going to have 1 less oxygen. Instead of 5, it's going to have 4. You can also think of ribose as having 1 more oxygen than DNA. Okay? All right, now this wheel right here tells us about the structure of using those nucleic acids, 3 in a pair, or sorry, 3 in conjunction, which we call a codon, to make an amino acid. So we're using these nucleic acids to eventually make these nucleic acids, these amino acids listed on the outside of this wheel. It's very important to understand how to read this wheel. You read this wheel basically from the inside out. And you basically follow, as we did in class, you follow from the inside out. So if I wanted to know how you make histidine, you start here, okay? And your first letter in the codon is going to be C, A, and then histamine is this way, so it's U or C. That means there's two different codons that are going to make this amino acid. Okay? So again, we went through this in class, but just so that you remember, following this wheel from the inside out is going to give you a codon, a series of 3 of these letters. The order of these 3 letters of the codon makes a difference, because it's not the same thing to say UCU as opposed to CUC. You make what? You make a completely different amino acid. Okay? Now, your proteins. Your proteins are, like I said, we went over this already, but your proteins are the amino acids or the amino acids joined together to make a protein. And they have a particular function. Your proteins, like I said, control the rate of reactions in the body. They help to fight disease. They regulate, what goes in and out of the cell. But their number one job is to build structure. Now, why is that important to mention? Because remember, what are proteins made of? They're made of linking hundreds and thousands of those amino acids together to form what we refer to as a polypeptide chain. So let's analyze an amino acid here for a second. You have 3 parts to an amino acid. You have your amino group, you have your carboxyl group, and you have your side chain in the middle. Side chain is also referred to as your variant, your R group, your reactant. The amino group and the carboxyl group, the amino group and the carboxyl group in all amino acids is always going to be the same. The thing that differs is always going to be here, the side chain, the variant. So as you can see, if you analyze these 2 amino acids, you will see that your amino group is the same in both. You will see that your carboxyl group is the same in both, and the only difference that you have is going to be your side chain. Okay? Oops. Your side chain. Okay? Now, what happens when we want to link these amino acids together? The thing that links amino acids together are going to be your peptide, okay, also known as a peptide bond. Amino acids linked together are joined by a peptide bond. The carboxyl group of one amino acid is going to join the amino group of another amino acid by a peptide bond. Okay? Eventually, when you start linking hundreds of these together, you get what is called a polypeptide, or we're going to refer to it as a polypeptide chain. So, again, I mentioned this earlier, but the amino acids are joined together by a polypeptide chain, and just remembering that amino groups and carboxyl groups are always the same in them. We have more than 20 different amino acids found naturally. Our body also helps in the assistance of amino acids as well. Where the amino acids differ is in the side chain. This is basically a picture here to give you the structure of an amino acid. I just kind of gave you this picture so you could see it kind of larger, but it's the same thing that we were just previously talking about. Now let's talk about the levels of organization. And we did this in class regarding the levels of organization, the 4 levels of organization in an amino acid. Sorry, in a protein. For protein synthesis, our first level is going to be referred to as our primary structure. Now basically what you have there is you have amino acids linking together, and here you have your peptide bond right there, which I kind of wrote over it. So there's your peptide bond, and here are your amino acids. Okay, they link with a peptide, and eventually this link is going to form a long chain. So that is basically your primary structure. It is the sequence or the linking of amino acids. Eventually they get to the point where they're going to start to fold or coil. You can see the coiling start already. This right here is actually occurring inside of it right there. Okay? Now, this along with other substances that are surrounded around it, you get what is called a secondary structure. Okay? You get a secondary structure when these amino acid chains start to fold one over the other. Once that folding continues, you get a tertiary structure. And a tertiary structure is basically where you have your 3D. You no longer have a top and a bottom, but now you have a top, a bottom, and a side. So now you have your three-dimensional arrangement, your tertiary structure. Okay? After that, this tertiary structure and that tertiary structure and a third and a fourth and a fifth and so forth start to link together. You now have the quaternary structure. Okay? Your quaternary structure is basically proteins built together, or your protein. So a protein with more than one chain is that fourth level of the structure. You have your polypeptide chain that starts to link. There's your primary. It starts to then fold. There's your secondary. It continues the fold until it develops that 3D arrangement. There's your tertiary. And then you start linking several of them together, and you end up with a protein. Okay? So for example, the protein hemoglobin consists of 4 tertiary structures. This is another picture to show the same thing to you. You have your primary structure, your polypeptide chain. Your polypeptide chain will start to fold, forming your secondary structure, or it'll start to coil as well. Okay? When it does that, it will continue and continue and continue to fold and coil to build your tertiary structure. Take this tertiary structure and another one and another one and another one, put them all together, you get your quaternary structure, which is basically your protein. Okay? Primary structure is your first sequence of amino acids. Your secondary structure is your amino acids that twist and fold. Your tertiary structure is the chain itself as it's folded, and your quaternary structure describes the different protein chains basically hook up together. Your different tertiary structures all basically hooking up together to make your protein. This is a chart here to show your 20 amino acids, just so that you would be able to see what they look like. You don't really need to know this exactly for the test. I just thought I'd provide it for you. Now let's talk about enzymes. We talked about how proteins serve to control the rate, not control the rate, but rather assist in the rate of chemical reactions in the body. Well, now we're talking about enzymes then. So enzymes basically speed up the process of chemical reactions in the body. That means that enzymes serve as what we call a catalyst. Okay? Enzymes are a catalyst. A catalyst is what we would basically say it's a substance. The enzyme is a substance that alters the rate of the chemical reaction. And basically what it does is it speeds it up. Now, how does it speed it up? Well, chemical reactions need what we refer to as activation energy. The energy needed to activate the chemical reaction, to make the chemical reaction happen faster. So when you look at this, let me clear this again. When you look at this, you'll see 2 reactions. You see one reaction with an enzyme, which is your black line, and you see one reaction without an enzyme, which is your red line. Now, as I did this in class, for periods 2, 4, 6, if you basically started these off together, the one without the enzyme requires more activation energy. That basically means it takes longer to occur. It takes longer to complete the chemical reaction. Therefore, if you could think of this line, your starting point and your ending point, this travel is going to take longer to occur. I can't do it on it, but it's going to take longer to occur than this. Okay? It's basically, think of it as a mathematical concept. This is a shorter line than this if I were to take the two lines and fold them out. So what I want you to get from this is simple. When you add a catalyst, when you add an enzyme to serve as a catalyst to a chemical reaction in the body, it is going to lower the activation energy required to make the reaction occur. By lowering the amount of activation energy, it will make the reaction happen faster. As opposed to if you have a chemical reaction occur without an enzyme, without the assistance of a catalyst or an enzyme acting as a catalyst, then the reaction takes longer to occur because the reaction has to utilize a greater amount of activation energy. Okay? So, with an enzyme, less activation energy. Without an enzyme, more activation energy. Now enzymes, like I said, are proteins, and they act as a catalyst. That means that they increase the reaction, they make it happen faster, they make chemical reactions happen faster in the body, like for example, in our digestive system. Enzymes contain what we call active sites, and substrates are going to react with those active sites. They're going to attach to them. So here you have your picture of your enzyme, and this is your active site right here. Substrates, these are our substrates, okay, are going to attach to the enzyme in a perfect little fit here. And then they form a bond to the substrate. They can also break apart. So this enzyme is going to attach the substrate to it, and it could actually move this to a different part of the cell, and it can create what we call a product by breaking the substrate off of the enzyme afterward. Okay? It's going to basically move it from one part of the cell to the other. Again, I put this picture back up because, again, this is in that reaction that we were just talking about with enzymes and substrates to produce a product. There's where your activation energy comes into play. The fact of bonding to that substrate requires energy, okay, to make a product. If you do it with an enzyme, you're going to create or you're going to utilize less activation energy. So what are some of the things that can affect the work of an enzyme? There are 5 things that can affect the work of an enzyme, and they're listed here. Number 1, the concentration of the enzyme itself. Secondly, the concentration of the substrate itself has an effect on the work that the enzyme will do. Also, more importantly, temperature. Enzymes work slower in colder temperatures, and they work faster in higher temperatures. This is partly because as temperatures increase and temperatures go up, in other words, if it gets hotter, the molecular motion increases and it creates collisions. Okay? So the heat is going to basically cause the enzyme to lose its shape, okay? And it's going to lose the hydrogen bonds that hold it together. Colder temperatures, on the other hand, are going to slow it down. So it's going to decrease molecular motion, okay? So higher temperatures increase enzyme activity, where colder temperatures slow them down. Your pH. We're going to talk about pH when we do properties of water, but your pH, basically, when we're talking about whether something is basic or acidic, also has an effect on the activity of an enzyme. When the pH levels are changed, your hydrogen and hydroxide levels are also going to be changed as well. So these fluctuations change how the enzyme basically folds. It changes then the shape of the activation site where the substrate can attach. And then the concentration, finally the concentration of salt. Each enzyme has what we call an optimal salt level, the perfect amount of salt concentration that the enzyme has. If you change that, you're going to change the behavior of the enzyme itself as well. Now, we also, let me just mention real quick that meat tenderizers contain an enzyme, like in this example, papaya and salt itself, they contain an enzyme that digests proteins. These tenderizers come from papaya. All right. So in other words, I wanted to also point out that enzymes, can serve good purposes, especially a natural enzyme like the one found in papaya as well. All right. At this point in the PowerPoint, we would now be doing the project with the project with the dots candy, and that concludes our Macromolecules 2 presentation.